Okay, so welcome back to this next video in which we are discussing atherosclerosis. Okay, so in this video what we're going to begin with is our discussion of the pathogenesis of atherosclerosis. Now it's fair to say that this is not one of the best understood processes in the world, but our information about it is growing. So I will present to you the model that we currently have, but it does have holes within it, okay? And it hasn't got everyone convinced. Okay, so let's have a look at what this model is then. So, basically the model that I'm going to present to you is what's known as the response to injury hypothesis, okay, or the response to injury model. And basically the idea is that atherosclerosis is the response to an injury within the endothelial cells. Okay, and that's where we'll start, with some injury to the endothelial cells that line the lumen of the artery. Okay, so let's start off by drawing these endothelial cells. Okay, so let's have endothelial cells here sitting on their basement membrane. Okay, so here we have two endothelial cells. Here are their nuclei, and at the moment we've got a healthy endothelium. Okay, so this isn't an endothelium where we're going to get atherosclerosis at the moment. Okay, but we're going to do the first trigger that actually causes the atherosclerosis now. So this at the moment is a perfectly healthy endothelium. Okay, and then what you're going to get is some sort of injury to this endothelium, and this is going to cause the dysfunction of these endothelial cells. Okay, right, so what sort of injury are we talking about here? Well, hemodynamic injury is a major factor, okay, and we know that because in places where you get atherosclerosis, generally it's places where the pressure that the blood must exert on the wall is greater. So for instance, a common site where you find atherosclerosis occurring is the endothelial cells that line the posterior aspect of the aorta within the abdominal cavity. Okay, so let me explain this in a little bit more detail. Where shall I draw this? Okay, so I was going to draw my next stage here, so I have to put it over here. So basically, in your abdominal cavity, you have the abdominal aorta moving down, basically, okay? So this has come from the thoracic aorta up here, which has then come out of the heart. And in the abdominal cavity, the aorta sits directly in front of the vertebral column. Okay, so sitting behind it then, you have the vertebral column. So this is the vertebral column, and sitting in front of it, we've got the abdominal aorta. So I'll colour the abdominal aorta in so it's completely distinguishable. So here's the abdominal aorta. And basically, a very common site for you to get atherosclerosis is in the endothelial cells which line the blood vessel just in front of the vertebral column. Okay, and this we think is because the blood, whenever the heart beats, is, you know, the aorta engorges whenever the heart pumps the blood into it. And we think what happens is the endothelium gets crushed against the back of the vertebral column here. And that's a very common site for atherosclerosis. So the posterior abdominal aorta here. Okay, right. So the pressure that the blood puts on the endothelial cells is a major factor. Okay, and this means that if you have chronically high blood pressure, known as hypertension, this could be a major risk factor for atherosclerosis. In fact, we know it's a major risk factor for atherosclerosis. Okay, which is why doctors are so worried about people who are hypertensive, because of the damage that it could be doing to their endothelial cells and the atherosclerosis that this could cause. Okay. In addition, other things that are known to cause it is if you have far too high LDL levels within your blood. The too high LDL can also damage these endothelial cells in ways that aren't fully understood. Okay, we'll see the involvement of LDL again later in atherosclerosis. It's going to play a huge role in this, not just in this initial injury. Okay, that's not understood. We're going to see a whole other role for LDL within atherosclerosis. Okay, but it also plays a role in this initial starting off of the whole thing, in that it can cause the dysfunction of these endothelial cells. Other examples of things which can injure endothelial cells are horrible chemicals that could be within your blood. Okay, so how are you going to get horrible chemicals within your blood? Well, for instance, if you smoke, you're always inhaling foul chemicals into your lungs, and then many of these chemicals will just go straight through the lungs, through the pneumocytes which line the alveoli, and then into the blood. Okay, so you're getting horrible chemicals into your blood. Okay, so the chemicals within cigarette smoke. Okay, and also obviously other smoke as well. You know, it's not good to be inhaling things out of power stations and other factories, which is why they have to have the huge, great chimneys so that it goes high above us all. Okay, right. So, anything else that I'd like to mention as far as injury? Also things like potentially bacterial infection, viral infection of these endothelial cells. People have looked and found tantalizing hints that potentially atherosclerosis is caused by infection. We'd absolutely love to find that. It would be like gastric ulcers or peptic ulcers rather. Peptic ulcers where for years and years and years, doctors, whenever they had someone with a peptic ulcer, they had to talk to them about their stress levels and things like that. And then some geniuses in Australia found that there was a bacteria that was causing it all, and now we can just dose these people up on antibiotics and send them away. So that's improved that. But we'd love to find that atherosclerosis is caused by a bacterial infection so that we can just dose people up on antibiotics and hope that they get better. But although there are tantalizing hints, and you do find bacteria within atherosclerotic plaques, we don't think yet that it is caused by a bacterial or a viral infection, but potentially they could be the initiators. Okay, they could cause the initial endothelial damage. In addition, irradiation can also lead to endothelial damage. Okay, right. So, what's going to happen then when these endothelial cells become injured? Well, basically they're going to do what they would do in an inflammatory response. Okay? However, should endothelial cells that are lining an artery be undergoing the reaction that endothelial cells undergo in an inflammatory response? In an inflammatory response, you've got some potential infection in your tissue, and what happens is the endothelial cells contract down to much smaller sizes, okay? And that opens up gaps between neighbouring endothelial cells, which then allows things in the blood to leave the blood and go into the tissue. And also they become very sticky and start grabbing onto white blood cells and moving them out of the blood into the tissue fluid. But that happens in capillaries and also post-capillary venules. It doesn't happen in massive great arteries, okay? So we're going to do what you do in inflammation, but in a massive great artery, basically. So what's going to now happen is these endothelial cells are going to contract down and they're going to start recruiting leukocytes. Okay, so when they contract down, they open up these massive gaps between them. Okay, so they increase vascular permeability hugely, and this means that things within the blood can leave the blood and go into the layer of the artery wall that's underneath the basement membrane. And what is that layer? Well, it's the subendothelial connective tissue. Okay, so the next layer was this layer that previously I coloured in red, which was the subendothelial connective tissue. Okay, or the subendothelial space was that other name that I gave you for it. So subendothelial space, I think I'll call it now. And it's part, remember, of tunica intima. Okay, so we've had permeability go up. Now, what permeability going up is going to mean is that LDL molecules can now actually sneak between the two endothelial cells here into the subendothelial space. So here comes an LDL molecule, and this is completely out of proportion. I have drawn the LDL molecule the same size as these endothelial cells. Remember, the LDL molecule was around 20 to 25 nanometers, much, much smaller than an endothelial cell. You can see endothelial cells down a microscope, a light microscope. You won't be able to see low-density lipoproteins down a light microscope. Okay, so I'm drawing it that big so that I can actually draw it, but in reality it will be a blip on this picture. Okay, so this is 20 to 25 nanometers. Okay, now let's just revise its structure. We have this phospholipid monolayer here with our massive great apolipoprotein B100, which has shrunk since the last picture, but never mind. Okay, and then we've got our phospholipids in the outer layer, our free cholesterol in this outer layer, and our cholesterol esters in the lipid core there. Okay, so LDL molecules come out of the blood and into the subendothelial space. In addition, these endothelial cells, which are undergoing the inflammatory response, they're going to start sticking to all sorts of inflammatory cells within the blood, and they're going to start moving them into the tissue. Now, we're going to see several of these later on, but for now, the main one we're going to be interested in is monocytes. Okay, so let me draw a monocyte here. So monocytes are so named because they have a massive great nucleus, okay? So they're often called mononuclear cells, okay, because the nucleus dominates them. And also, you're comparing them to other leukocytes, which just is the fancy word for white blood cells, such as the neutrophils and the eosinophils, which have multi-lobed nuclei. And compared to those, the nucleus of the monocyte is extremely boring, and therefore it's called the monocyte, because it's only got a single lobe, basically. Okay, right, so here is our monocyte, and it's now stuck to our endothelial cell. And basically, monocytes are the precursors to macrophages, so you do not have macrophages, which are these great blobs of cells involved in the acute inflammatory response and also in the initiation of the adaptive immune response. Okay, you do not have those circulating within the blood. Instead, you have the precursors to macrophages circulating within the blood, and the precursors to macrophages are these monocytes. So the monocyte will then be moved through this opened gap between the endothelial cells into the subendothelial space here. And when it gets there, it will differentiate into a macrophage. And macrophages are great blobs of cells here. So this is a macrophage. Okay, so these dysfunctional endothelial cells have firstly opened up and allowed LDL molecules to leave the blood and accumulate in the subendothelial space. And worse than that, they've brought these monocytes in with them as well. So they've moved monocytes from the blood into the subendothelial space. And they've become macrophages. So they've differentiated into macrophages. They've changed their pattern of gene expression. Okay, right. So now, what do the macrophages start doing? Well, you have brought these macrophages in. Why have you brought them in? What is the body doing here? Well, it thinks that there is something absolutely terrible happening within the subendothelial space. That's why these endothelial cells have contracted like this. They're bringing in cells that are designed to come in and obliterate intruders, okay? And that's why people are really looking for any sort of infection that can be linked to atherosclerosis, because it really does look as though the body thinks there is some infection occurring within the subendothelial space, okay? But we haven't really found that evidence yet. The leading sort of idea is chlamydia pneumoniae, okay? But it's sort of, you can't find it in everyone. It's a little bit, it's contentious still. Okay, so at the moment I'm not going to include that there, but you might like to think that the body is launching an attack as though there is a horrible pathogen present within the subendothelial space. So these macrophages are coming in here, and what are they supposed to do? They're supposed to obliterate whatever is in here that shouldn't be in here. Okay, now, what do they do to do this? Well, one of the things that they do, which is the significant thing that they do as far as the development of atherosclerosis is concerned, is that they're going to secrete truly horrible molecules. Okay, and there's one truly horrible molecule that we're going to discuss in particular. And this horrible molecule is known as superoxide. Okay, and it's within the larger class of molecules known as reactive oxygen species. And for short, reactive oxygen species are often abbreviated to ROS for short. So these can be abbreviated to ROS. Now, what is the structure then of superoxide? Well, to see this, let's start by looking at the structure of oxygen. So let me draw a nice big picture for the structure of oxygen then. So oxygen molecules consist of two oxygen atoms bound together by a double bond, like so. And this double bond contains two single covalent bonds. And in both of these single covalent bonds, you have two electrons, one from each member of the bond. Okay, so this bond contains one electron from this oxygen and one electron from this oxygen, and this bond here also contains one electron from this oxygen and one from this oxygen. Okay, so this is an oxygen molecule here. Now, to convert oxygen molecules like so, which are sometimes called dioxygen molecules, okay, because they've got two oxygen atoms within them, to superoxide, what you need to do is fire a single electron, which I'll colour in black. This is the bad electron, okay? It's going to come in here, and it's going to break the... I've just realised how racist what I just said could be taken to. I'm sorry about that. I've coloured this electron in dark, just arbitrarily. Okay, right. This bad electron is coming in, and it's going to split one of these bonds within this dioxygen molecule here. Okay, so one of the bonds is going to break, and what you're going to create is an oxygen atom linked by a single covalent bond to another oxygen atom. Okay, like so. And this electron will go to one of them. Okay, so let's say it goes to this oxygen over here. Okay, now let me just complete this diagram here, because both of these oxygens are also going to have two electron pairs. Okay, like so. So here are two electron pairs on this oxygen, and here are two electron pairs on this oxygen here. Okay, what's now going to happen is we're splitting this bond up. This electron will go back to this one, and this electron will go back to this oxygen. And I'm going to say that this electron here is going to go to this oxygen here. So what's going to happen is this oxygen here will end up now with 3 lone pairs of electrons. It'll also end up with a negative charge because it's received this electron here as a new separate electron which had a negative charge on itself. So it's now got a negative charge here. Meanwhile, this oxygen over here, what's this going to end up with? It will have 2 lone pairs of electrons from here, and then this is the baddie. It's got 1 electron that is on its own here, and electrons do not like being on their own. This is the free radical, okay? And when you have electrons on their own in molecules, that's known as a free radical, okay? So that's what this is all about. And basically these are incredibly reactive structures. No matter what you're talking about, if you have an unpaired electron, a free radical, it will be incredibly reactive. These are dangerous, dangerous chemicals. Okay, so the macrophage starts producing these superoxide molecules. And for short, superoxide is often denoted as O2, because you've still got 2 oxygen molecules, well, 2 oxygen atoms rather. Then you've got a negative charge on one of these oxygens, and then a free radical on the other, and people put a dot afterwards to denote the free radical. Okay, or if you wanted to draw this, you might just draw it like so. One of them has a free radical, the other has a negative charge. Okay, so we are producing these superoxide molecules. Now, why is the macrophage spinning out these superoxide molecules? Well, the idea is that we have some incredibly dangerous pathogen potentially in here. At least that's what the body is reacting as though you have. Okay? And we are going to poison this pathogen out. So the macrophages come in and they start releasing these poisons, which will just go and react with anything that they come across, basically. So if you have a pathogen in the presence of these horrible molecules, they're going to get a full dose of them, and these molecules will react with their proteins, they'll react with their lipids, they'll react with their DNA, they will destroy them, basically. Okay, so the macrophages are coming in, releasing all of these horrible reactive oxygen species with the hope of destroying the pathogen. However, the pathogen either isn't there, or maybe it is and we just don't understand it yet. But the thing is, even if it is there, you're going to do more damage than good, potentially, by releasing these horrible superoxide molecules. Because what's now going to happen is that superoxide is going to interact with the LDL molecules. Okay, and it's going to convert these LDL molecules from something that's okay, it's a physiological thing, into something that's horrible. Okay, so superoxide is going to come in and react with the lipid molecules that are in the LDL molecule. It will react with the protein that is in the LDL molecule, the apolipoprotein B100. It will react with anything else that I haven't mentioned, which is in the LDL molecule. Okay, and then what you get is this modified LDL molecule that has reacted with superoxide and has undergone more chemical reactions than can possibly be listed, basically. And we now know it as an oxidized LDL molecule. Okay, so if you're wondering what oxidized LDL actually is, well, the answer is we don't really know. We don't know which of the modifications that the superoxide has made to the LDL molecule is significant for what this molecule will now do. Okay, the superoxide reacts with everything basically in here and produces all sorts of modifications, but we don't know which of these is the significant one. Okay, so oxidized LDL literally just means LDL that has reacted with superoxide molecules. Okay, right. So, in fact, I'll draw this in a different colour now. Okay, I'll draw this in vivid purple. So this is an evil LDL molecule now. Okay, so here is our LDL that has reacted with superoxide and is now called oxidized LDL. Okay, right. And for short, oxidized LDL is often abbreviated to OX-LDL for short. Okay, right. So it's been modified in all sorts of ways. It might be the modification to the protein, the apolipoprotein B100, that is significant, or it might be the modifications to the lipids. We don't know. But this molecule is now different after having been doused in superoxide molecules that have come from the macrophages. And now what's going to happen is these macrophages are going to say, wait a second, this stuff is damn dangerous. Okay, we better engulf that and get it out of the extracellular fluid. We don't want cells coming into contact with this, so we'll engulf it and spare other cells from coming across this stuff. Okay, so what happens is the macrophages have a receptor on their surface which interacts with the oxidized LDL molecules. Okay, so here's our blob of a cell that's a macrophage. Okay, here's its nucleus. And they have a receptor which is a scavenger receptor on their surface, like so. So this receptor here is a receptor within the family of scavenger receptors. Okay, and what will happen is this will lead to them uptaking the oxidized LDL molecule, and what they will then do is they'll break down the oxidized LDL molecule, they'll break down the apolipoprotein B100, they'll break down all of the phospholipids that made up the phospholipid monolayer, they'll release the free fatty acids, and they will use those for their metabolism potentially. Okay, and then what they'll be left with is a huge number of cholesterol esters and also cholesterol molecules. Okay, they will break down the cholesterol esters into a free fatty acid and a cholesterol molecule, and what that then leaves them with is an absolutely huge supply of cholesterol. Okay, so let's try and squeeze this in here. So these macrophages, basically by engulfing these oxidized LDL molecules, are giving themselves an absolutely massive great dose. Whoops, I've drawn that as a 6-membered one rather than a 5-membered one. They're giving themselves a massive great dose of cholesterol, basically, and it's potentially oxidized cholesterol. Okay, so massive great dose of cholesterol. Now, the uptake of this oxidized LDL triggers the activation of the macrophages further, okay? And what they're going to do is they're going to secrete tumor necrosis factor alpha. Is just one of the many things that uptaking the oxidized LDL is going to trigger the release of. Okay, so basically what happens is these macrophages come across this oxidized LDL, they say, goodness, that shouldn't be here, that's something terrible, and then they say, we're going to need backup to deal with this. They then start sending out alarm signals, one of which is tumor necrosis factor alpha, TNF alpha, and I should, I suppose, write the full name of this out. So this stands for tumor, that's the N, the T rather, and I'm sorry if you're an American and want the different spelling, just cross the U out. The N then stands for necrosis, the F stands for factor, and then it's tumor necrosis factor alpha. Now, there are different tumor necrosis factors other than the alpha one. However, tumor necrosis factor alpha is easily the most important of the tumor necrosis factors. So if anyone ever just says tumor necrosis factor without clarification as to which tumor necrosis factor they're talking about, you can assume they are talking about tumor necrosis factor alpha. It's the most important one. Okay, so some people will just call this TNF. Okay, and what will happen is the TNF will go back to these endothelial cells up here, okay, and it will cause further activation of this inflammatory response. So you're going to get positive feedback of this entire thing. You're going to activate more of these endothelial cells to do this silly behavior, basically, where they're recruiting monocytes and opening up gaps, okay? So it's going to make the whole thing become this vicious cycle, basically. Okay, right. So we'll call it there for this video, and in the next video what we'll discuss is other effects of this interaction with this oxidized LDL and how it's going to take us towards atherosclerotic plaques.